Hello everybody.
Morning, ma'am.
Good morning. Okay, so today I will take the class of placenta. Now the placenta, you know that it is present. It is a transitory organ and it is connecting the developing fetus to the uterine wall, isn't it? So it is a temporary organ which connects the developing fetus by the umbilical cord to the uterine wall. Normally it is connected to the upper part, that is on the posterior wall, but sometimes it may be connected to the anterior wall. Sometimes may be anywhere, or it may be attached just above the opening of the uterus, that is known as the internal os, and that condition is known as placenta previa. Whatever, the normal diameter of this placenta is about 15 to 25 centimeter, and the thickness is about 1 inch, and weight, so nobody usually weighs unless the research is going on the placenta. So its weight is about half kilo, that is 500 to 600 gram. And that placenta, it is an important organ for the fetus because it provides nutrition, that is the carbohydrate, the amino acid, fatty acids, okay, vitamins, all these things it will be to the fetus it will provides. And it also eliminates the waste product and also helps as a lung of the fetus, because the lung in the fetus doesn't work, so the oxygen, the gaseous exchange also occurs between the fetal blood and the placental, that is the maternal blood. Okay, and also it protects fetus. What, how? Because the antibody, that is that immunoglobulin G, I think you know about that immunoglobulin G. So it is it goes from the maternal blood to the fetal blood throughout fetal life, and this fetus cannot develop this immunoglobulin G unless he or she is 3 years old. Okay, so the immunoglobulin G, that is the antibody which protects the fetus from any infection, from infections. Okay, then it produces many hormones. First of all, in the early pregnancy, it produces an hormone that is known as human chorionic gonadotropin, and then it secretes progesterone. The human chorionic gonadotropin is secreted the progesterone is secreted by the corpus luteum of pregnancy, and this human chorionic gonadotropin, it protects that corpus luteum of pregnancy. It helps in growth of the corpus luteum of pregnancy, which can secrete progesterone, and that progesterone helps to maintain the thickness of the uterine wall, and also prevent the muscular contraction during pregnancy in the early part. But later on, that placenta itself, it secretes progesterone in the early part of fetal life, and then at the end of pregnancy it again produces some hormone, that is the estrogen, which increases the muscular contraction, that helps in muscular contraction, just opposite of the progesterone action. And also it helps in the development of the mammary gland during pregnancy, as well as that after pregnancy also. So that is not by the placenta, because at birth the placenta will be delivered. Usually 30 minutes after the delivery of the baby, the placenta is delivered. So that is about the placenta. Now this is the, in the placenta, when you see that, you will see it has a shiny surface, that is fetal surface, and a rough surface, brown in color, that is maternal surface. And in the shiny surface you will get, why it is shiny? Because it is covered by a membrane, and that membrane is the amnion. And on the fetal surface, the umbilical cord is attached. Usually it is attached to the midline, slightly eccentric. So slightly eccentric position, the umbilical cord is attached. Whereas if you see the maternal surface, you will see that it is very rough, and there are grooves are also present. In between the grooves there will be elevated area. That elevated areas are known as lobule, or cotyledons. Cotyledons or lobules. And there are about 15 to 20 cotyledons are present in the placenta. Okay. Now you should know about the how the placenta is developed. Okay. Now if we go from the ovulation time, so after ovulation, the ova will enter into the fallopian tube, and then at the ampulla of the fallopian tube, if sperm comes, then it will be fertilized and form the zygote. And after that formation of zygote, soon it starts to divide, and that is, it will form the 2 cells, 4 cells, 8 cells. So when it is 8 cells, then it will enter into the uterine cavity, that is in the day 4. Gradually there will be some changes, some fluid will appear, the cells will be divided into embryoblast and the trophoblast outside, and there will be a cavity known as the blastocele, and the whole thing is known as blastocyst. Now this blastocyst is now implanted at about 8th through 9th day after that ovulation. It will be implanted by proliferation of the trophoblast, and these trophoblastic cells, they soon proliferate. They cannot make the wall, they cannot get time to make a wall surrounding them. So there will be only the cytoplasm, and the nuclei are present in the cytoplasm, so that is known as syncytiotrophoblast. But in the deeper part of the syncytiotrophoblast, the trophoblast, they will form the cell boundary, so they are known as the cytotrophoblast. Okay. Now on 9th day, when it is implanted, you can see that there will be a gap, and that gap will ultimately covered by a coagulum, fibrin coagulum. Okay. And this dark green, it is the syncytiotrophoblast, and the light green is the cytotrophoblast, deep to the syncytiotrophoblast. And here you can see that embryoblast, that is that the germ cells, they will be divided into 2 layers, that is epiblast and the hypoblast, and 2 cavities are formed, that is amniotic cavity and the yolk sac cavity. And you can see that the lacuna has appeared inside the syncytiotrophoblast. So these are known as the trophoblastic lacuna. Whereas this is the uterine endometrium where it has been implanted. And here you can see the blood vessels, they are dilated. And these dilated blood vessels, also there are some other changes. The glands will be enlarged, there will be edematous fluid is there, and the nutrition, it is filled up with nutritious substance. So that type of endometrium is known as decidua. So it is implanted in the decidua. The blastocyst is implanted in the decidua. Now the lacuna has appeared, and now that syncytiotrophoblast, they will proliferate soon, and they will enter into the blood vessels. They will erode the blood vessels wall, and as a result, as they erode the wall of the blood vessels, dilated maternal blood vessels known as the maternal sinusoids, so they will now pour blood into this lacuna, the trophoblastic lacuna. So lacunae are filled up with blood, maternal blood. Now here you can see, after some time you will see that cytotrophoblast is also proliferating, and it is surrounded by the syncytiotrophoblast, and that is a solid finger-like structure, and that is known as the primary villi. So here you can see the primary villi. This is the cytotrophoblast surrounded by the syncytiotrophoblast, and surrounding that there is the trophoblastic lacuna filled up with maternal blood. Now gradually the mesoderm will appear. The mesoderm will appear deep to the cytotrophoblast, that is known as the extraembryonic mesoderm, also surrounding the yolk sac and the amniotic cavity. Okay. and here you can see that the primary villi is formed. Now this mesoderm then that will enter into this primary villi and it will form the secondary villi. Later on, the blood vessels will be formed in this mesoderm and that will also be entered. That is the fetal blood vessels that will also be entered into that mesoderm, and that will form the tertiary villi. Okay, so here is the primary villi, that is solid cord, finger-like projection, and here you can, this is the trophoblastic lacunae and they are present in between the villi, so that is known as the intervillar space, which are filled up with the maternal blood. And then the mesoderm will enter, and after some time, that is at the end of the third week. So primary villi appear at the beginning of the third week, and the secondary villi appear in the middle of the third week, and the tertiary villi, that is when the blood vessels will enter, that is the third part of the third week. That is the later part of the third week. Okay, so during third week, all these chorionic villi or the placental villi are formed. So that is the fetal part, that is fetal part of the placenta. Whereas the decidua will form the maternal part of the placenta. This decidua is known as the decidua basalis. Now, after some time, we know that there is the cytotrophoblast, outside there is the syncytiotrophoblast, and there is the mesoderm, and inside the mesoderm the blood vessels are present, that is the fetal blood vessels. They will form the capillary, they will divide into branches, ultimately form the capillary. Now the cytotrophoblast, it grow faster and it comes close to the endometrium. It comes close to the decidua and it is attached to the decidua. Now that is known as the outer cytotropoblastic shell. Now this type of villi is known as the stem villi or anchoring villi. And from this stem villi, that some villi they will come out and they will float into the maternal blood vessel. So they are known as the floating villi or branching villas. Branching or floating villas. Okay. Now this is the area where the embryo is connected with mesoderm. Embryo is connected with that placental region by the mesoderm known as the connecting stalk. Okay. Now by fifth week, here you can see that the tertiary villi are appeared and they appear all over. Now there are three cavities. Here I have not shown that one. In this region, the mesoderm will appear, and in the mesoderm there will be cavity will be formed, and that cavity is known as the chorionic cavity. So there will be the amniotic sac, there will be the yolk sac, and there will be the chorionic cavity. And the chorionic cavity, there is the mesoderm surrounding it, and also this is the connecting stalk which is connecting the developing embryo to the placental region. So this is the connecting stalk. Okay. Now here this is the anchoring villi. Now here you can see that the tertiary villi, they are more in number, they are growing in size, but it is also appear surrounding the whole of the embryo in a radiating manner. Gradually, only near the embryonic pole, this is the embryonic pole, here these villi, they will grow more and more, but on the other side, these villi, they will gradually degenerate and they will ultimately disappear. Okay. Now here you can see that this is the uterine cavity, which is this is the myometrium outside the uterine wall, myometrium, and here that is the endometrium, that yellow color is the endometrium, and that this is also endometrium here where the embryo is implanted. Now here you can see on the embryonic pole, the villi, they will grow more and they will form like a tree-like structure, and that is known as chorionic frontosum. chorionic frondosum. Okay, and on the other part, the villi will be degenerated, and that part of the chorion is known as chorion levy. That part is known as the chorion levy. Okay. Now, where the chorion frondosum is present, there the decidua is known as the decidua basalis. Where the decidua is present in this degenerated area, that is known as the decidua capsularis. Decidua capsularis. And the decidua which is present in the rest of the uterine wall, that is known as the decidua parietalis. Now that amniotic cavity, that chorionic cavity and the embryo, they will grow and grow and grow. Ultimately, what will happen, that this is the decidua parietalis, and this is the decidua capsularis surrounding the chorionic levy, and the decidua parietalis, they come close to each other, and ultimately they will fuse, and the uterine cavity will be obliterated. There will be no uterine cavity. Uterine cavity will be obliterated. Okay. So here you can see that this is the chorionic frondosum. This is the decidua basalis. This is the decidua capsularis covering the chorionic levy, and this is the decidua parietalis due to growth of the embryo and the amniotic chorionic cavity, and the amniotic cavity also covers the whole of the embryo, and it also come close to the mesoderm, and the amniotic mesoderm come close to the chorionic mesoderm, and the chorionic cavity also disappears. So only the amniotic cavity is present, and the uterine cavity also disappears. The uterine cavity, because there is fusion of the decidua capsularis and the decidua parietalis. So the uterine cavity will also be disappear only in the lower part. And here we can see that amniotic membrane and the chorionic membrane, they are fused with each other, and they will form the amniochorionic membrane. And during delivery time, this amniochorionic membrane, it will come close to the uterine opening, that is known as the internal os, and it will rupture, and the amniotic fluid comes out. Okay, so that is known as watering. So water will come out, the amniotic fluid will come out as if the water is coming out through the vagina. Okay, so that is obliteration of the uterine cavity. Now here you can see the placenta is formed. So placenta is formed by two structures. One is the chorionic frondosum, another is the endometrium. That is, endometrium is the decidua basalis. So the placenta is formed by two structures. One is the maternal part, one is the fetal part, and the maternal part, that is the decidua basalis, and the fetal part, that is the chorionic frondosum. Okay. Now you can see that that cord is attached. This cord is developed from the connecting stalk, and that cord is known as the umbilical cord. It is usually attached to the middle, but slightly eccentric also. Okay. So that is at about 4th month, the uterine, 4th to 5th month, the uterine cavity will obliterate. Okay. Now you can see the placental structure. So this is the chorionic frondosum, fetal part, and this is the maternal part, that is decidua basalis, isn't it? Decidua basalis. Now here you can see that the the decidua basalis by about 5th month or second half of pregnancy, this decidua basalis, they starts to send septa. They start to send septa. Okay, in between the villi. They start to send septa in between the villi. These septa, they are known as the decidual septa. These septa, they are known as the decidual septa. But they do not touch the chorion, that is the mesoderm. They do not touch the chorion or the mesoderm. So this intervillous space, this is one villus, this is another villus. So this intervillous space, they will communicate with each other. So maternal blood is present in the intervillous space. And this is the one villus is present and they divide the placenta into lobules or cotyledons. They divide this decidual septa. They will divide the placenta into lobules or cotyledons. Each cotyledon consists one chorionic frontosum, and here you can see that stem villi is present, the floating villi is present. Inside there is the fetal artery and the fetal vein. The fetal artery that carry the deoxygenated blood, because it reaches placenta for oxygenation. So the fetal artery, like the pulmonary artery, it also carries the deoxygenated blood, and the fetal vein that will carry oxygenated blood. The fetal artery carry the deoxygenated blood, and the fetal vein will carry oxygenated blood like the pulmonary artery. And here these are the uterine artery, endometrial artery. They are known as the spiral artery, and this spiral artery, because they are very much tortuous and very much dilated, and inside this artery the cytotrophoblast also enters and it replaces the endometrium. So they are dilated, they cannot contract. Okay, they can contract but not so easily. They are dilated, very much dilated. And there are about 80 to 100 spiral arteries are present which pour blood into the intervillous spaces. Whereas here, these intervillous spaces, their pressure is more, about 80 to 100 millimeter of mercury, and that will go deep to the villus space. And from there, the oxygen by diffusion that will be entered into the fetal vein. Okay. Then that fetal vein, here they have made it blue, but actually it should be red because it carries oxygenated blood, and that fetal blood then enters into the fetus. Then from the fetus, again the deoxygenated blood comes through the umbilical artery and it enters into the chorionic villi, and ultimately it will be drained into the endometrial vein. So here the cotyledons you can see like this. These are the groove which is formed by the decidual septa, and here the chorion is covered by a thin layer of decidua basalis. Okay, so that is the placental structure and circulation I have told. Now what is the placental barrier? Now this is the chorionic frontosum or the tertiary villi where the fetal capillaries are present. If we take a cross section, then we can see that these villi are lined by the cytotrophoblast, and then outside by the syncytiotrophoblast. Inside there is the mesoderm and the blood vessels. So what will be the placental membrane or the placental barrier? Here is the intervillous space. So these are the layers: the fetal vascular endothelium, then the connective tissue or the mesoderm, then the cytotrophoblast, then the syncytiotrophoblast. But this happens only in the early part of pregnancy. There are 4 layers. Then after fourth month, what will happen? The cytotrophoblast, they degenerate. Here these are the cytotrophoblast. They degenerate. The 5 number, that is cytotrophoblast. They will degenerate. And mesoderm will also be degenerated. As a result, the fetal capillaries, they will come close to the syncytiotrophoblast. So now the placental membrane, here you can see these are the fetal blood vessels, and this is the syncytiotrophoblast, and the fetal blood vessels come close to the syncytiotrophoblast. So the placental barrier is formed only by 2 layers. One is the endothelium, another is the syncytiotrophoblast. So though there is placental barrier, but that placental barrier can be crossed by many viruses like rubella viruses or toxoplasmosis bacteria, or many other teratogenic organ. They can pass through the placental barrier. Though there is a barrier, but still the structure can pass through this barrier. This barrier is not very effective barrier. Okay. Now, the placenta can be classified according to the maternal chorion interface. What is that? Now, in human being, if we see in human being, and if you see that this is the fetal capillaries, this is the fetal capillaries, that is the endothelium, and there will be the syncytium, that is the placental barrier here, and this is the intervillous space. So the capillary is very close to the maternal blood, so that is known as hemochorial type. The chorion, this is the chorion, and this is the that is chorionic villi, and that this is the blood vessels in the chorionic villi. So they are very close to the maternal blood, and that is known as hemochorial type of placenta, and that is very invasive type. That blastocyst or the embryo, it is present inside the endometrium, inside the uterine wall of the uterus. So it is very invasive type. Another, it is found in the tigers, lions, dogs, cats. So the other carnivores, it is present. So there is syncytium, and there is no intervillous, there is intervillous space, but that intervillous space is lined by endothelium. So it is moderately invasive, So that is in carnivores. But in ruminants or in the herbivores, like in the cow, goat, horse, pig, so they they that just touch the uterine endometrium. So uterine epithelium is also there. So after delivery, there is no damage to the uterine epithelium. So this is the maternal capillary, this is the fetal capillary, in between there is the uterine epithelium is also there. So that is known as epitheliochorial type of placenta, and it is the non-invasive type of placenta. So in the full term placenta, that will covers about 15 to 30% of the internal surface of the uterus. Okay, and their diameter already I have told, that is 15 to 25 centimeter, thickness is 2 to 3 centimeter, and weight is about 500 to 600 gram. Okay, so after birth, that model is there. So that placenta model, usually it is kept in the exam table. So you will tell that what is fetal surface, what is maternal surface, why there are groups, what are the elevated areas in between the groups. The examiners want to listen that you will tell it is cotyledon, how many cotyledons are there, why these groups are formed. These groups are formed due to the decidual septa. And what is maternal surface? So there are cotyledons covered by a thin layer of the decidua basalis, and this is it is a smooth surface, shining surface, transparent surface. Through that transparent surface one can see, but in the model that smooth surface is made white. Okay, in our department that model is there, that is the smooth surface or fetal surface of placenta is white, but actually it is so transparent. Through this transparent part you can see the red placental surface. Okay, that is maroon placental surface you can see, and the umbilical cord is attached here, and the blood vessels are going towards that umbilical cord, isn't it? And here will be the amniotic membrane. This is the amniotic membrane. The white layer will be the amniotic membrane, the transparent membrane, that is the amniotic membrane. So I think that is the last slide, and if you have any questions about this class, you please ask. Then the assignment is that you have to describe a human placenta, that is what are the features of that human placenta, and then you have to show how it is formed, that structure, and how the blood is circulated through this placenta. So that is the assignment. So I think you have no problem. So thank you very much. Thank you.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Now placenta, placenta, you know that placenta, it means an type of cake, like the chocolate cake. And what type of cake? Strawberry cake, isn't it? So it is a type of cake. It is a religious type of cake. It is in the ancient Greek and Roman, they used to take this cake That is the placenta. The placenta means cake. Okay. Thank you.